Bismillahirrahmanirrahim. Assalamu alaikum. I welcome all my students to today's lecture of GIT. And what is that? That is the movements of the small and large intestine. Now we are going to talk about the movements taking place in the small gut first of all. Now always keep one thing in mind is that there is presence of two types of movements in the small gut, and what are they? They are the mixing contractions and the propulsive contractions. The mixing contractions, they are actually the contractions so that the food should be properly mixed with the secretions inside the gut, whereas the propulsive contractions, they are responsible for movement of food downward or head towards anus. Now, essentially, all the movements of the small intestine cause at least some degree of both the mixing and propulsion as well. Now, mixing contractions, or you can say the segmentation contractions, when a portion of the small intestine becomes distended with chyme, stretching of the intestinal wall elicits localized concentric contractions spaced at intervals along the intestine and lasting a fraction of a minute. The contractions, they cause a segmentation of the small gut, and they divide the intestine into spaced segments that have the appearance of a chain of sausages. See, the segmentation contractions, they are regularly spaced. They can be isolated, they can be irregularly spaced, they can be weak, regularly spaced. Now, as one of the segmentation contractions relaxes, a new set often begins, but the contractions this time occur mainly at a new point between the previous contractions. The segmentation contractions, they chop the chyme 2 to 3 times per minute, in this way promoting progressive mixing of the food with secretions of the small intestine. The maximum frequency of the segmentation contractions in the small intestine is determined by the frequency of electrical slow waves in the intestinal wall. The maximum frequency of the segmentation contractions in duodenum and proximal jejunum areas is about 12 per minute under extreme conditions of stimulation. In the terminal ileum, the maximum frequency is about 8 to 9 contractions per minute. The segmentation contractions become exceedingly weak when the excitatory activity of the enteric nervous system is blocked by the drug atropine. The slow waves in the smooth muscle itself that cause the segmentation contractions, these contractions, they are not effective without background excitation, mainly from the myenteric nerve plexus. See? Now let's talk about the propulsive movements. Peristalsis in the small intestine. The chyme is propelled through the small intestine by peristaltic waves. These can occur in any part of the small intestine, and they move toward the anus at a velocity of 0.5 to 2 centimeter per second, faster in the proximal intestine and slower in the terminal intestine. They are normally weak and usually die after traveling only 3 to 5 centimeters, rarely further than 10 centimeters, so forward movement of the chyme is very slow. This means that 3 to 5 hours they are required for the passage of chyme from pylorus to ileocecal valve. Control of peristalsis by nervous and hormonal signals. Peristaltic activity of the small intestine is greatly increased after a meal. This is caused partly by the beginning entry of the chyme into the duodenum, causing the stretch of the duodenal wall. Peristaltic activity is increased by the so-called gastroenteric reflex, which is initiated by distension of the stomach and conducted principally through the myenteric plexus from the stomach down along the wall of the small gut. In addition to the nervous signals that may affect small intestinal peristalsis, several hormonal factors also affect the peristalsis. And what are they? They are the different hormones like gastrin, cholecystokinin, insulin, mortallin, serotonin, all of which enhance the intestinal motility and are secreted during the various phases of food processing. Secretin and glucagon inhibit small intestinal motility. On reaching the ileocecal valve, the chyme is sometimes blocked for several hours until the person eats another meal. At that time, a gastroileal reflex intensifies the peristalsis in the ileum and forces the remaining chyme through the ileocecal valve into the cecum of the large gut. Now let's talk about the propulsive effect of segmentation movements. The segmentation movements, although lasting for only a few seconds at a time, often also travel 1 centimeter or so in the anal direction and during that time propel the food down the intestine. Now, at the same time, we should be knowing about this term peristaltic rush. We should be knowing about what is peristaltic rush. Peristalsis in the small intestine is normally weak. Intense irritation of intestinal mucosa, which occurs in severe cases of infectious diarrhea, can cause both powerful and rapid peristalsis, known as the peristaltic rush. It is initiated partly by the nervous reflexes that involve the autonomic nervous system and brainstem, and partly by intrinsic enhancement of the myenteric plexus reflexes within the gut wall. Movements which are being caused by the muscularis mucosae and muscle fibers of the villi. The muscularis mucosae can cause short folds to appear in the intestinal mucosa. In addition, individual fibers from this muscle extend into the intestinal villi and cause them to contract intermittently. The mucosal folds increase the surface area exposed to the chyme, thereby increasing absorption. Contractions of the villi, shortening, elongating, and shortening again, so that the lymph flows freely from the central lacteals of the villi into the lymphatic system. So, draw and label the villi is important. Then we should be knowing about the functions of ileocecal valve. Now, its function is to prevent the backflow of fecal contents from colon into small intestine. Ileocecal valve itself protrudes into the lumen of the cecum and therefore is forcefully closed when excess pressure builds up in the cecum and tries to push the cecal contents backward against the valve lips. The valve usually can resist reverse pressure of at least 50 to 60 centimeter of water. See, the ileocecal valve made by the sphincter, and the pressure and the chemical irritation relaxes it, and the fluidity of contents promotes the emptying, The pressure of or irritation in the cecum inhibits the peristalsis of ileum and excites the sphincter. The wall of ileum for several centimeters immediately upstream from the ileocecal valve has a thickened circular muscle, so the word sphincter has been added to it, and this sphincter normally remains mildly constricted and slows emptying of ileal contents into the cecum. Immediately after a meal, a gastroileal reflex intensifies peristalsis in the ileum, and emptying of ileal contents into the cecum proceeds. Resistance to emptying at ileocecal valve prolongs the stay of chyme in the ileum and thereby facilitates absorption. And normally only 1500 to 200 ml of chyme is going to empty into the cecum each day. So this is all about the today's lecture, and inshallah we'll continue later on. Stay blessed. Allah Hafiz.